Hello everyone. Today's topic of discussion is isomerism in carbohydrates. So what is the reason for this isomerism in carbohydrates? It is the presence of asymmetric carbon atom that imparts isomerism. Here you can see the structure of glucose. There are four asymmetric carbon atoms in glucose. We can see in C2, C3, C4 and C5, four different groups are attached to the carbon atom. So there are four asymmetric carbon atoms or chiral carbon atoms. It is the presence of these asymmetric carbon atoms that imparts isomerism. Isomerism can be of two types. First is the stereoisomerism and second is the optical isomerism. Remember, it is the presence of chiral carbon atoms that imparts these properties. Is there any relationship between number of asymmetric carbon atoms and number of stereoisomers possible? Yes, that relationship is called Lebowitz-Hoff rule. It shows the relationship between the number of asymmetric carbon atoms and number of stereoisomers possible. So number of isomers possible is 2 raised to n, where n is the number of asymmetric carbon atoms. Remember, in 2 raised to n is the number of asymmetric carbon atom and not number of carbon atoms in the structure. So when you take the structure of glucose, there are 4 chiral carbon atoms. So number of stereoisomers possible is 2 raised to 4, that is 16. First, let's discuss about stereoisomerism. Stereoisomerism can be of 4 different types. First one is functional. That is functional isomerism, where there are different functional groups, whether it's a keto group or the aldehyde group. Second one is enantiomerism. That is the capital T, capital L isomerism, where there are mirror images. Third is anomerism. Anomerism with a functional group attached to the C1 in case of aldose and C2 in case of ketose. There is a difference in orientation of H and OH group around the anomeric carbon atom. And fourth one is epimerism, where there is difference in orientation of H and OH group around the carbon atoms other than the anomeric and penultimate carbon atom. So these are the 4 different types of stereoisomerism we will discuss in detail. First, let's see enantiomerism. So enantiomers, they are the type of isomers, stereoisomers, in which one isomer is a mirror image of the other. This requires presence of an asymmetric carbon atom. So capital D, capital isomerism is a type of enantiomerism. As you can see, capital D glucose and capital L glucose are enantiomers. In D glucose, the OH group is on the right side, and in L glucose, the OH group is on the left side. Functional isomerism is another type of stereoisomerism. Here, different functional groups are attached. That is glucose and fructose. These are functional isomerism. In glucose, it is aldehyde group, and in fructose, it is keto group. The third type of stereoisomerism is anomerism. So what is the reason for this anomerism? It is a formation of ring structure in monosaccharides that results in the creation of additional asymmetric carbon that is called the anomeric carbon. The carbon atom with the functional group forms the anomeric carbon atom. So in case of aldoses, the anomeric carbon will be C1, and in case of ketoses, the anomeric carbon will be C2. So the difference in orientation of H and OH group around the anomeric carbon atom results in anomerism. So the resulting isomers are called alpha and beta isomers. Examples of anomerism is alpha D glucose and beta D glucose. Similarly, alpha D fructose and beta D fructose. It is a difference in orientation of the H and OH group around the anomeric carbon atom that results in anomerism. context, let's discuss about mutarotation. So it is the process of interconversion of alpha and beta anomers in aqueous solution. So mutarotation, it is studied by measuring the rotation of plane polarized light. The optical rotation of alpha D glucose is plus 112 degree and the optical rotation of beta D glucose is plus 19 degrees. So both undergo mutarotation over a period of few hours and an equilibrium is attained. So at equilibrium, the optical rotation is plus 52 degrees. The fourth type of stereoisomerism is epimerism. So epimerism is the difference in orientation of H and OH group around carbon atoms other than the anomeric carbon and penultimate carbon. So the difference in configuration is confined only to one asymmetric carbon atom. As you can see in this picture, here you can see the second epimer of glucose is mannose and the fourth epimer of glucose is galactose. So we just discussed that D-mannose is second epimer of glucose and galactose is fourth epimer of glucose. But what about mannose and galactose? They are diastereoisomers. So we know that the mannose and galactose, they differ in configuration of more than one asymmetric carbon atom, right? So they are diastereoisomers. Whereas mannose and glucose, they are epimers, but mannose and galactose are diastereoisomers. So we just discussed about stereoisomerism. So there are four types: functional isomerism, then enantiomerism, anomerism, and epimerism. These are the four types of stereoisomerism. Now let's discuss about optical isomerism. So when a beam of plain polarized light is passed through a solution of carbohydrates, it rotates the light either to the right or left. So depending on the direction of rotation, two optical isomers are possible. That is, when they rotate the plane polarized light to the right, then it is called dextrorotatory, that is plus. So when the plane polarized light, when they are rotated to the left or the anti-clockwise, then it is called levorotatory. So this dextrorotatory and levorotatory, they are represented by small d, small l. So D-glucose, it rotates the plane polarized light to the right, that is clockwise. Hence, it is dextrorotatory, which is represented by small d or plus. So D-glucose is dextrorotatory. So that is why glucose is also called dextrose. Similarly, D-fructose is levorotatory. It rotates the plane polarized light to the left, that is anti-clockwise. And levorotatory is represented by small l or minus. So D-fructose is levorotatory. So why is sucrose called an invert sugar? This is because sucrose is dextrorotatory. That is, on hydrolysis of sucrose, it gives glucose and fructose. Though glucose is dextrorotatory and fructose is levorotatory, the levorotation of fructose is stronger. So, because of strong levorotatory nature of fructose, sucrose on hydrolysis, it becomes levorotatory. So that is why it is called invert sugar. That is, initially it was dextrorotatory, but on hydrolysis, it gives glucose and fructose, and the levorotation of fructose is stronger, so the sucrose becomes levorotatory. So that is why sucrose is called an invert sugar. Also, you should know about racemic mixture. The equimolecular mixture of optical isomers where there is no net rotation of plane polarized light is called a racemic mixture. So we just discussed about the two types of isomerism seen in carbohydrates. The first one, stereoisomerism, and second is the optical isomerism. Stereoisomerism can be of four types, that is functional, then D and L, that is enantiomerism, then anomerism and epimerism. And optical rotation based on the rotation of plane polarized light. It can be dextrorotatory or levorotatory. So any doubts from you?
Ma'am, I have a doubt. can you explain the difference between distereoisomers and epimers?
Epimers differ in orientation of H and OH group only to one asymmetric carbon atom, that is other than the penultimate and anomeric carbon atom. But in case of diastereoisomers, they differ in variation in position of H and OH group, that is seen in C2, C3 and C4. As you can see, mannose and glucose, they are epimers because mannose and glucose, they differ in orientation of H and OH group in second carbon atom. Whereas mannose and galactose, they differ in orientation of H and OH group in C2 as well as C4, right? So they are diastereoisomers. So when the difference in orientation of H and OH group is confined only to one asymmetric carbon atom, then they are epimers. But if there is more than that, then they are diastereoisomers. Is that clear, Nikki?
Yes, ma'am. Thank you so much. Ma'am, I have a doubt. Can you explain the difference between capital D capital L isomerism and small d small l isomerism?
Capital D capital isomerism are enantiomers. That is, they are mirror images. Okay, like D glucose, capital D glucose and capital L glucose. So they are just exactly mirror images. Whereas small d and small l, they are optical isomers. That is, they differ in orientation of, they rotate the plane polarized light either to right or left. In case of small d, that is dextrorotatory, they rotate the plane polarized light to the right, and L levorotatory, they rotate the plane polarized light to the left. Okay, so small d, small l, they are optical isomerism, that is dextrorotatory and levorotatory, whereas capital D, capital L are enantiomerism, which is stereoisomerism, where they are mirror images. Is that clear, Lepi?
Yes, ma'am. Thank you so much. So capital D capital L are enantiomers, which are mirror images, and small d small l, they are optical isomers, right? That is dextrorotatory and levorotatory.
Yes, Lepi, you are right. Very good. Now, can you answer this question? Which of the following are enantiomers? D-galactose, L-glucose, then D-galactose, L-glucose, D-mannose, L-mannose, and D-mannose and L-mannose. What is the answer?
Ma'am, it's D mannose and L mannose. See, capital D mannose and capital L mannose.
Yes, you are right, Nikki. Very good. Now, Lepi, can you explain me why?
Yes, sure, ma'am. These enantiomers are mirror images, that is capital D and capital L. So in case of first option, it is capital D galactose and capital L glucose. So they are not mirror images, so that cannot be the answer. Second option is small d galactose and small l glucose. So that small d small l are optical isomers and they are also not mirror images. So that is not the answer. Third option is capital D mannose and capital L mannose, which are exactly the mirror images. So they are enantiomers. And the last option, small d mannose, small l mannose, they are optical isomers. So here the correct option will be C.
Exactly. Very good, girls. Now answer this question. Which of the following are epimers? D-glucose, D-fructose. Then second option, D-mannose, D-talose. Third option, D-glucose, D-mannose. And fourth option is D-glucose and D-glucose. What is the answer?
Ma'am, answer is C. D-glucose, D-mannose. Mannose is the second epimer of glucose.
Yes, exactly. Very good. Can you explain other options too?
Yes, ma'am. Here in this case, only the third option, D-glucose and D-mannose, they differ in orientation of H and OH group in one carbon atom other than the anomeric and penultimate carbon atoms. That is, mannose is the second epimer of glucose. Other options do not match the criteria.
Yes, good. Now you can see in this picture, these are the various stereoisomers of glucose. So you can see the second epimer of glucose is mannose and fourth epimer of glucose is galactose. But in the question, the options given were glucose and glucose. We know that they differ in orientation of H and OH in more than one carbon atoms. Okay. Similarly, mannose and talose, they also differ in orientation of H and OH group in more than one carbon atoms. So they are not epimers. Epimers, they differ in H and OH group in one carbon atom. Okay, other than the reference and the anomeric carbon atom. Okay, so these are the epimers of glucose. So second epimer of glucose is mannose, fourth epimer of glucose is galactose, and third epimer of glucose is allose. Okay, so epimers differ in orientation of H and OH group in one carbon atom. Whereas diastereoisomers, they differ in orientation of H and OH group in more than one carbon atoms. Okay, is that clear, girls? So that's all about our discussion regarding isomerism in carbohydrates. I hope you're clear with the topic. Thank you, girls.
Thank you, ma'am.